Hey folks, and welcome to another edition of Zog Science. Today we're going to be taking what we learned in part one, looking at the theories of energetics, and we're going to look at the applications of what we actually see happening in real cells. So ATP is what is going to power all cellular work, and it's going to do so by coupling exergonic to endergonic reactions. Basically what that means is that ATP, when it is broken down into ADP and phosphate, is an exergonic reaction. That free energy that is released is going to be used to power endergonic reactions that are going to be working against entropy, or that are going to be decreasing entropy. There are kind of three different types of work that are done. We have chemical work, which is where we use ATP to do some sort of endergonic reaction. We also can do transport work, so we can push things against the concentration gradient, or we can do mechanical work, which is things such as cilia, moving, pulsing to be able to move a cell around, or contracting of muscles, or moving of chromosomes, things like that. So ATP is the energy of choice for cells. That is adenosine triphosphate. You can see that it has a pentose sugar right here, and then it's got an adenine nitrogenous base right here, and then it's got three phosphate groups, hence the triphosphate. It is a very high energy state, and the reason why is that all of these negative charges on the phosphates are all pushing and repelling away from one another. So it is very unstable, and it very much wants to get rid of this phosphate in order to do other things. And in order to get to that point, it is hydrolyzed. So we add a water molecule in, it turns into ADP, and then the inorganic phosphate, the last one, is kicked off, and we are left with that free energy, which we can then use to do cellular processes. So how does it work? It works a lot of the times by phosphorylating one of the reactants in a reaction, thus making it unstable, and this instability drives the reaction to become exergonic. So if we were to take glutamic acid and ammonia and try to combine them together into glutamine, the delta G for that reaction is positive 3.4, and that means that it's not going to happen spontaneously. We have to add energy to it. So what we do instead is we take the glutamic acid, we attach a phosphate group from an ATP to it. Now this glutamic acid actually wants to bind with the ammonia. It is unstable as it is, and so the ammonia will actually bind to that and kick off the phosphate group. And if you take a look, the reaction of ATP turning into ADP and the phosphate, that has a delta G of negative 7.3 kilocalories. And so if we combine that together, the net reaction is negative 3.9 kilocalories. And so overall, this is an exergonic reaction because we have coupled the exergonic reaction of the ATP hydrolysis with the endergonic reaction of the glutamic acid combining with the ammonia. So this is the way that a lot of reactions work with ATP. We attach that phosphate group, we use that free energy in order to drive the reaction forward. Some other examples, membrane proteins work this way. So the ones that do some sort of active transport, a phosphate group is a lot of times attached to that, and that phosphorylates it, gives it that free energy it needs to do. That phosphate in this case is actually going to change the shape of the protein, and that makes it do its work. We can also have ATPs which will attach straight to like a vesicle that is attached to the cytoskeleton, and then once that ATP is hydrolyzed, that causes the protein to move down the skeletal tract and therefore move the vesicle around the cell. So a couple different ways, but it's the same overall idea. We start off with ATP, it goes through an exergonic reaction and helps to power some sort of endergonic reaction. The regeneration of ATP it is renewed through its own endergonic reactions. So those are going to be things like cell respiration, fermentation, and we're going to talk about those in more detail in the next video. But it is renewed through its own set of exergonic reactions. So you can kind of see where this is going, right? We have exergonic reaction leading to endergonic reaction, leading to exergonic reaction, so on and so forth. And this is, again, why organisms have to be open systems, because we have to be continuously adding free energy into the system. Now, ATP by itself can do a lot, but it can't do everything. And the reason why is that reactions are just going to kind of be happening at their own rate without something to kind of help speed them up. And so we have catalysts, which in a biological systems are enzymes that are there to help speed up the reactions. And enzymes are a type of protein and they basically are just there to help make sure that the reaction is occurring. When you are identifying acids, many times they have an ACE ending, and a lot of times they're going to refer to their substrate. So, for example, sucrose is broken down by sucrase, which is the enzyme that helps to facilitate that reaction. Some other enzymes are catalase. You might have remembered DNA polymerase from biology, and then there are just tons of them, tons and tons of enzymes, thousands upon thousands in your body. Now, the reason why we need a catalyst and or enzymes is that reactions require activation energy. So it might be that the products are less, have less energy than the reactants. However, we still have to get over the fact that that along the way it requires a little bit of energy to get started. And so that activation energy is usually coming from molecules just kind of running around and bumping into one another. Okay, that kinetic energy of them running around can be enough to overcome that. However, not always, right? A lot of times when they bump together, they might not be able to get enough energy to make the reaction go forward. So what enzymes do is that they decrease the activation energy, therefore making it much more likely that the reaction is going to occur. And without enzymes, complex life could not exist. Your mitochondria without enzymes would not be able to produce enough ATP for you to survive. It's just not possible. So it's pretty important that we have these enzymes. Now, enzymes have several characteristics, one of them being that they're specific. They only work with certain substrates. So sucrase only works with sucrose. DNA polymerase only works with DNA. And they only work with what we call substrates, and substrates are the reactants in their reaction. It's what we're putting in that their shape must bind to. Enzymes are also reusable. They're not affected by the reaction, so they can continue to catalyze many times over. So you don't have to have a lot of them in order to continue to push the reaction forward. You can just start off with a small amount. And then finally, they function best at certain pH and temperature ranges, salt concentrations, things like that. And the reason why, well, remember they are proteins, and so proteins are, have the alpha helixes. They've got the beta pleated sheets. They've got all the R group interactions, and so when we change the pH, temperature, salt concentrations, that just completely messes them up. It causes their shape to change, and they're going to denature. So this is remember the reason why membrane-bound organelles are so important is because it keeps the enzymes in their specific environments where they have the best conditions so that they can function optimally. Now, when a substrate binds to an enzyme, or substrates bind to an enzyme, they bind to what is known as the active site and what is called the induced fit theory. I'm sure many of you might remember the lock and key model. The induced fit theory is a little bit newer model. And basically what it says is that rather than it just being like a lock and key where the shape of the lock and the shape of the key don't really change, the substrate is actually more like a hand and the enzyme is a glove. And so what happens is that, this enzyme binds kind of around the substrate and changes its shape a little bit to help fit it, just like a glove when you put it on changes its shape a little bit to accommodate your hand going inside. So it's not quite the lock and key theory. It's a little bit different. There is a little bit of movement that can occur. And so that means that, when they're outside of their optimal range, they might still be able to work to some extent, but they just won't be able to work as effectively. So just kind of the process of how this occurs. Again, a substrate enters the active site or substrates. The enzyme changes its shape, binds to it. They're usually held in there by some sort of weak interaction like a hydrogen bond or like an ionic bond, maybe some sort of polar, non-polar interactions. The active site then lowers the activation energy and it helps to speed up the reaction. The substrates are then converted into their products. They are then released, and then the enzyme is able to go back to its inactive shape where it can bind with new substrates. Now, enzymes don't just function on their own. There are cofactors and coenzymes that can be necessary for proper function. A lot of vitamins and minerals, things like zinc, iron, vitamin A, those types of things, they're actually coenzymes and cofactors, and they help to bind to enzymes to help them to function properly. We can also have inhibitors. Inhibitors actually suppress enzyme activity, and they do so by binding. And they can either do that as a competitive inhibitor, where they bind to the active site, or they can be a non-competitive inhibitor where they will bind on another portion of the enzyme, but by binding to that other spot, they change the shape of the enzyme so that way the substrate cannot bind. These can be either irreversible or reversible inhibitors, and so that allows you to either have control, levels of control over how the reaction is going to occur. One of the types of regulation is called allosteric regulation, and this is where the active site might have the enzyme might have 4 different subunits. This might be the quaternary structure, and so an activator will bind and it will keep it in a stable active form, or we can have an inhibitor bind that would keep it from being able to function. We could also have another type of activation where when one substrate binds, it will allow all of the other active sites to be available as well. You don't necessarily need to know the specific names for these, but we do want to make sure that we understand the concept that enzymes can be turned on or turned off by other molecules rather than just the substrate. What this all allows is for us to have either positive or negative feedback regulations. So a lot of times what happens is that the end of a metabolic pathway will actually be an inhibitor to an enzyme along the pathway. So for example, in the isoleucine production pathway, we start off with threonine, which is the initial substrate. It goes through several different intermediates and enzymes. They all go through. They eventually form isoleucine. Isoleucine comes back and it binds to a non-competitive site on the enzyme, and it will prevent the enzyme from being able to bind with the threonine, thus cutting off the process. So you can imagine how this is very useful, right? All these things require energy, and organisms want to make sure that they are using the least amount of energy possible to sustain life. That's just efficiency. It's something that we'll see continuously throughout the whole year. So once you know you have enough isoleucine, the isoleucine can bind to one of those enzymes. Eventually, that's going to stop the production of the isoleucine. Isoleucine levels go back down. It disengages, and now you can produce it more again. And so this feedback loop helps to maintain efficiency within a cell. Finally, location, location, location. We talked about this in our last unit with cells, but I'm going to repeat it again. Enzymes are organized into organelles because you have more control over each reaction. Factors like temperature, pH, concentration of the substrate, salinity, all these things are very important. And so it's important to maintain these enzymes within their specific location, whether that's the mitochondria, the lysosome, peroxisome, whatever. And so the location is incredibly important, and that's why we have organelles, right? We talked about that a whole bunch. So thanks for watching, and I'll see you next time.